All right. Hi, everyone. Thank you so much for joining us today. We're so happy to have Dr. Aaron Shafer from Trent University presenting for us today on the genomic history of deer. Aaron Shafer is an associate professor in the Department of Forensic Science, Environmental and Life Sciences, and in the Applied Modeling and Quantitative Methods graduate programs. His research group uses molecular tools to reconstruct demographic events and to identify the molecular mechanisms underlying phenotypic variation. He's also interested in applying genomic methods to conservation and legal issues and serves as an associate editor for Conservation Genetics. So thank you, Dr. Shafer, for being here today. The floor is yours.
Wonderful. And everyone can hear me okay?
Yes.
Well, thank you for the invite. I would love to come to Guelph in person next time. Guelph's one of my favorite cities. I love the campus. But nonetheless, I'm happy to chat with everybody and tell you a story about deer. And just to begin, I wanted to acknowledge that I'm on the traditional territory of the Mississauga and Anishinaabe. And if you ever get the chance to come to Peterborough, you can visit the petroglyphs, which aren't far, and you'll actually see some deer carving. And that'll be a theme throughout this talk today, sort of human relationship with deer. And so I'm going to tell you work I've been doing for almost a decade now on deer. I'm going to divide it up into two parts: population genomics and quantitative genomics. The first part on population is really about the speciation and human history with deer. I'm not saying this is a Canada Reads story, but it's a story that I'm proud of. It's something I started at Trent, and I think it's follows the traditional arc of a story. And then I'm going to talk for the latter third a little bit about some quantitative work that we're doing. And I think this might be of interest to the CGIL group, but on methods we're applying to estimate heritability by circumventing pedigrees, and also some different ways we're going about gene mapping, trying to find the genetic basis of traits. But to begin, I wanted to give you a brief history of deer in North America. And so deer, when I say deer, I'm talking about cervids. And so that's deer, white-tailed deer, mule deer, elk, moose, caribou. So we have 5 species of deer in North America. But if you were on this landscape 10, 15,000 years ago, there were actually quite a few more species. And so if you follow the meat eater, they did a spotlight on the extinct deer of North America about a year ago. Stag moose is one of them. Mountain deer is one of them. And on the right here is the subway deer, which you may have heard about on the news recently. That was my lab. And we used ancient DNA to identify what we believe to be another extinct species of deer that was located here in North America. So, there are 5 species of cervid now, 4 of which are in Ontario. But, 10,000 years ago, there were considerably more. The 2 big ones of my research focus are on white-tailed deer and mule deer. White-tailed deer is what you have on Guelph campus and what we see in Ontario. Massive range that's shown here on the right. Mule deer is what we see out west, shown here on the left. And there are some key characteristics. Mule deer are also black-tailed deer. And so you can see they have a black spot on their tail. They have different antlers, very different habitat. Ears are another characteristic. If you live where they overlap, it becomes quite easy when I lived in Alberta to differentiate the two. But if you're not familiar with both of them, they can be confusing. And they do hybridize. Where they overlap, hybridization is relatively common, depending on the study you look at. And humans have had, especially in North America, a long history with deer. And so if you look at various archaeological studies, you will see evidence that certainly in Eastern North America, the first peoples often would have relied on white-tailed deer as a primary source of protein. And so this is just a highlight from a site where the vast majority of bones evidenced of what the communities were eating were actually white-tailed deer. So foodstuffs is important. Clothing and shelter shown here in the top right. The hides are of value in deer. And then in the bottom right here, I like to highlight this. This is actually when Champlain first made it into Southern Ontario 400 years ago. There's some translation considerations, but he referred to as the Indigenous community in Southern Ontario as the people who tend deer. And in fact, there's been some debate about historical tending and farming deer by First Nations in Eastern North America. It's likely what Champlain was talking about was deer drives, but nonetheless, it highlights that, you know, for tens of thousands of years in the continent, people have worked and relied on deer. When Europeans colonized, deer became actually the first example of a wildlife management law and this idea of hunting season. So in Rhode Island, just east of us, in 1646, this is the evidence of the first formal hunting seasons. And so the state enacted a law to prohibit hunting in particular seasons. And just to give you an idea as to how reliant Europeans were on deer in North America, here's one record of Savannah, Georgia. And so they were hunting deer and sending the pelts back to Europe. And so from just over the course of just over 15 years, 2.6 million pounds of deer skins from 600,000 whitetail deer were sent from Savannah, Georgia, to England. And this was happening all up and down the eastern coast of North America. So long history of First Nations living and relying on deer, and then Western Europeans coming over and enacting management laws, and this is the beginning of sort of massive deer exploitation on the continent. Because of this exploitation, both from hunting and land use changes in North America, and I think this is always surprising for people that don't study deer, deer were largely extirpated across North America. The data in Canada is a little bit more sparse, but so this book here on the left looks at the history of deer from 1878 to 2004, very detailed inventory of census data, but also stalking data. And what we saw right around the turn of the century was a complete crash in whitetail deer and mule deer, black-tailed deer in North America. So just south of us in Ohio, completely extirpated. And you know, now there's millions of deer. There's 30 million deer in the continent, but not too long ago, 130 million years ago, in this black line, you can see that the deer were extirpated from large portions of their range. You also see this recovery. And so after European colonizers largely extirpated deer, then what followed that was this massive effort to recover populations and translocate them. And so again, this stalking book here details the immense effort that wildlife managers took to move deer across the continent. We've done this in Canada as well, but not at the scale of the US. And we can see the numbers actually recovered relatively quickly. And I'm going to come back to this book because we rely on some of the data here to understand genomic patterns. So European exploitation resulted in a crash, but also a recovery. And in terms of modern day relationships with deer, they are definitely a part of our lives. There are property values in Ontario, but certainly in parts of the US that are impacted by the presence of trophy deer, big antlered deer. And so here's an acreage for sale in Kansas, and they spotlight large white-tailed deer as one of the highlights. With the number of deer and the human interactions, there's also diseases we need to be aware of. Chronic wasting disease is a big one. And this is 100% fatal. It's a prion disease. It's been detected. It actually was detected in Ontario, even though technically Ontario, we are not on the map. But at the Toronto Zoo, I think in the 70s, there was a positive case. But it's certainly in all of our neighboring jurisdictions. And so this is a big concern because it is fatal. And the prion, if you know things about prions, it's next to impossible to destroy in the environment. So it persists. COVID is actually also another consideration with deer. We've been doing deer captures, my group, with the ministry for 5 years now, and roughly about 5 to 10% of our animals will have COVID antibodies. And this is a STAR article from 2022, where work in Ontario actually also detected what appears to have been a deer to human transmission of COVID, because the variant that's circulating in deer is quite distinct. We also have overabundance of deer. So with this recovery, we have overabundance. So Point Pelee, just west of you guys, we'll actually close the park down in the winter to cull deer. And I work a lot at Thousand Islands National Park. and they just completed a cull. Very controversial. These are national parks, but the deer are considered overabundant and destroying the natural ecosystem, so they've taken this action. So lots of interactions between humans and deer, and hopefully you'll see this theme throughout my talk. And before I get into what we've been doing, I want to give you a bit of a primer on speciation genomics. And so one of the questions I've been interested in is trying to understand the history between white-tailed deer and mule deer. And so if you've taken first-year biology, you would have learned about speciation and probably Ernst Mayr. And so this was really the idea of islands and allopatry being the proximate cause of speciation. But what genomics has allowed us to do, and this sort of traces back to really Darwin's idea of natural selection, was this ability to look for speciation that occurs while gene flow is taking place, and kind of placing a premium on adaptation rather than allopatry. And so I've been particularly interested in trying to tease apart these two modes of speciation in deer with genomic data. And genomic data, you don't need to know a ton about genomic data, but know that we get a lot of information, and there are summary statistics and patterns that emerge. And it turns out there are some patterns that if we compare the genomes of two species, if you look at these scenarios on the left, there are 3 different summary statistics based on their relationship. We know that elevated FST and DXY, you don't need to know what these are. No, we can generate them from genomes. That pattern is associated with divergence with gene flow. So species that separate but continue to exchange genes. And we can compare this to allopatric selection. That leaves a very distinct signature on the genome, which is the Mayr model, and background selection, which is sort of our null model. And so using genome data, I can compare white-tailed deer and mule deer, for example, and see what their genomes look like and do they fall into one of these patterns. And we can also determine on the right here how far along they are on the speciation continuum. Are they good species, quote unquote, when we look at their genomes. When we do these genomic models, one thing you just need to be aware of is we need to know rates. Rates in particular are mutation and recombination rates. And so when we do genomic studies and we try to understand divergence, often underlying the models is a number associated with mutation rate and recombination rate. And I just wanted to flag this because this isn't to be taken lightly. We often just use default numbers when it comes to mammals, but a really cool study came out last year in Nature on whales, showing essentially because they were using the wrong mutation rate, they overestimated human exploitation. They were applying a much faster rate than actually what was determined to be, and this as a result suggested that the crash due to whaling was much more severe than it was. And so you can actually estimate mutation rates using trios, mother, father, offspring, shown here on the left. You can estimate germline mutation rate. We've done that in deer. And so as I mentioned, I've been capturing deer for 5 years now. And often we will capture mother, father, offspring. And when you do that, you can identify the number of new mutations in the offspring that essentially appeared in the sperm or the egg. So those are germline mutations. And then we can get an actual rate. And so just to highlight here, this is a cool thing we're doing is when we talk about mutation and recombination rates, all of those in my lab are coming directly from the species we study. We did the same thing with recombination. We were able to estimate recombination, and now we're actually sequencing. I won't talk about it today. We're actually sequencing sperm cells to identify germline recombination. And so some neat things we're doing. And so all of our models where we try to understand speciation and demography are based off of deer values, which I think is pretty cool. And so when it comes to deer speciation, white-tailed deer and mule deer, what do we know? This is essentially the working model of speciation in deer, and it's based off mitochondria. And what it suggests is over time we've had secondary contact events. So the species diverged 3 million years ago, and then maybe 2.5 million years ago, they overlapped, exchanged genes, separated, and this cycle repeated. So this is this idea of speciation with gene flow. And based off mitochondrial, this gave me a sort of our null expectation that when we look at the genomes, I would expect to find a history of the species separating but exchanging genes, so speciation with gene flow. So to go about addressing this question, my lab has been sequencing genomes for almost a decade now, and we have probably 150 different deer genomes spread across their range, some high coverage, some low coverage, some from island populations that have cool things about the species, a lot of data. And so this is what I'm working with. And we're always interested in samples from South America. People think of white-tailed deer as only being in North America, but the white-tailed deer actually expands into Central and South America. And so I'm very keen to get samples from that part of the world. Now, to look at deer speciation, you can do this a few ways. One, you can just look at the genomes and generate a PCA. How do the genetics cluster? When we do that, what we see on the left-hand side is we have 4 groupings of deer, and they largely align by species. And so white-tailed deer across the range are in the top right. So that's all of essentially North America. White-tailed deer form one genetic cluster. But the key deer, which are a unique population found in the Florida Keys, are separate from their white-tailed deer in the white-tailed deer group. Then in the left side, we also have mule deer and black-tailed deer separating from each other, but also white-tailed deer. Admixture plots on the right suggest no evidence of recent gene flow in our samples. This is not surprising. Genetically, they are very distinct species, and we can even identify major populations, or some people might even call them subspecies. When we try to look for evidence of historical gene flow, we can do this a couple ways. I wish I could see everybody because I usually ask this question. But one of the ways is through what's known as the ABA-BABA test. And so if you have 4 populations, 2 species, up here on the left, your normal phylogeny or tree should be AA, BB, or BB, AA. I'm trusting everybody's nodding when I say that, but that's the null expectation. When you get patterns, when you look at their DNA that come out as BABA or ABA, the only way you get that pattern, well, there's 2 ways, but one of the ways you get that pattern is through historical introgression, historical gene flow. And so when we're trying to test whether or not species have exchanged genes in the past, ABA-BABA is a very valuable test. And if the value deviates from 0, that would suggest that there's been historical gene flow. Now, when we compare the relevant populations in the red box, everything's essentially 0. So the ABA-BABA test says no evidence of historical gene flow. This is counter to what the mitochondrial tree would suggest. We can do some more complex demographic models here on the right. Everything in the dashed line would represent when the 2 populations I'm comparing split. So if we just look at the bottom, this is white-tailed deer, the bottom 2, white-tailed deer and mule deer. And the blue bars are evidence of migration. We have virtually no evidence of migration or gene flow after the species split. We would expect blue bars to be on the left side of that dashed line. Nothing would suggest historical gene flow. We often do more complex demographic models. This is sort of akin to what you see in humans when we have these very complex scenarios. We can do the same thing in deer. Again, what I just want to highlight is this M value. The arrows are not to scale. They're relative to one another. We're detecting essentially no migration between the 2 species. And so in complex demographic models shown here, none of them support this idea of speciation with gene flow. Rather, we're seeing a signal consistent with allopatry. Mule deer and white-tailed deer were separated and didn't exchange any genes. We can also look at this continuum again following this pattern, and the relevant group to look at is in the bottom right here. If we compare between species to within species, between is on the bottom right, within is on the bottom left. We have this negative correlation between recombination and FST. These are 2 summary statistics that we expect to be negatively correlated. And the stronger the correlation is what's observed when you're far along in the speciation continuum. So when we do that, we see that white-tailed deer and mule deer, despite hybridizing, and we know they hybridize currently, they're actually quite far along in the speciation continuum, and we should consider them as good species. Well, when we look at their mitochondrial data in our analysis, we see that same pattern of the species model where it's a complete mess, more or less, where mitochondrial data for both species are not clustering by species. So purple and red here are each individual species. You can see they're forming intermixed clades. And so the mitochondrial data is highly consistent with the pattern of what we would call incomplete lineage sorting. And just for reference too, so the species don't form mitochondrial clades. The subway deer, which we just published last month, is embedded within this clade, and also Otocoileus pandora, which is the brocket deer found in Mexico, is also found within this clade. So it's a highly discordant mitochondrial pattern, not consistent with species, not consistent with geography, and something we think is likely a result of incomplete lineage sorting. And so to summarize that part, we see no evidence of gene flow during speciation. And the pattern of the mitochondria is either due to ancestral hybridization or incomplete lineage sorting. And I tend to lean towards the latter. And so very clean nuclear phylogenies or nuclear genomes, no signal of gene flow. Mitochondrial, it's the exact opposite. When we look at more recent demographic history, we can do all sorts of cool modeling. Maybe you've seen these plots, but we can model their effective population size over time and how things change. And what I wanted to highlight here with respect to the purple lines, that's white-tailed deer, is there was a massive impact of the last glacial maximum. And this makes sense if you remember what their range looked like. They covered essentially all of North America right now. But 20,000 years ago, the majority of their range was covered by ice. And we pick that up in their genomes, and we see a massive demographic collapse that corresponds to the last glacial maximum. In contrast, everything in green is the mule deer and black-tailed deer. They've been low and had a low effective population size for hundreds of thousands of years, and consequently were less impacted by the last glacial maximum. We can also pick up the recent impact of humans in the genomes, which is a really cool thing we're able to do now. So again, everything in purple in the top box is the genetic effective population size. And you can see roughly about 150 years ago, we see a massive drop in the effective population size of white-tailed deer. And one of the cool things we did was we extracted all of the data from that stocking book, and we superimposed it over these patterns. And so in the bottom plot here, I'll break this down for you. So the solid lines are just the census size, and you can see they perfectly correspond with the pattern we're seeing with the genetics. Every dot reflects the low in any given state. So if we look at the top, there's a black dot and there's a gray line and it says AL, that's Alabama. So all of these dots reflect the recorded low in any given state, and that perfectly coincides with the genetic drop. And then the gray bars are the length of time each state spent restocking and bringing animals in. And what we can see again is that those gray bars perfectly overlap the genetic recovery. And so it's a cool exercise where we're able to actually pick up these very recent events, largely caused by humans, in the genomes of our deer. And if you're curious, when you have a massive drop like this, it may or may not affect diversity. And it turns out that it actually doesn't affect genetic diversity in white-tailed deer. So we looked at we've been doing ancient DNA. And so these 4 dots here on the left are diversity pre-crash, and every black triangle is the diversity after the crash, and there's no difference. And so what often happens in bottlenecks is if you're in the bottleneck, but you get out of it quick, the impact on diversity is pretty minimal. And we see that in our data. And so we're able to pick up this recent demographic history. We're also able to measure the cost of some of these demographic patterns. And so one of the things we're interested in measuring, and from a livestock perspective, this is fairly common, but measuring the genetic load. And what we see in white-tailed deer, if we compare them to caribou and mountain goats, they have a much higher genetic load than other ungulates on the landscape. And this is actually expected. white-tailed deer have very high diversity, and we know most species that have high diversity also have a high genetic load. They just tend to mask it through heterozygosity. And this genetic load isn't distributed equally amongst deer. So in this plot on the right, we have mainland deer, but we also have, for example, the Florida Keys, which is considered an endangered species. They have an actually a considerably elevated genetic load relative to an already high genetic load in the population. And so we talk a lot about masked load, which can be hidden by heterozygosity, and realized load. And we speculate that the realized genetic load in the Florida Keys is actually relatively high. And so there's been some demographic and genetic load cost to this recovery. And one of the cool things we did was we actually tried to model it. And what we see here in green was we modeled each state that we had data for, and we looked at whether or not the number of animals that they restocked and the number of states that they used impacted diversity. And so here the prediction would be more states, more animals, less of an impact. And we actually see that. So the places that brought in more deer from more places, that had a negative effect on inbreeding, shown here in green, and in some of these genetic load metrics. And in fact, in red, and what these plots are, we also see that when the more recent the stalking event ended, so the last time the populations that we were fiddling around with most recently also have the highest genetic load. So on the y-axis here is time, the x-axis is, say, inbreeding or deleterious alleles, and we see this positive relationship. And so this is indicative of, okay, these populations were most recently in trouble, and we actually pick that up with their genomes. And it's worth highlighting this pattern because we pick it up in a species like white-tailed deer, where the cost and consequences of translocating animals might be minimal. But when it comes to isolated populations, this can actually be problematic. And so we know, for example, in the island fox story off the coast of California, these populations were thought to be locally adapted. And so actually translocating animals can actually be potentially harmful, breaking up co-adapted alleles and maybe counterproductive to conservation strategies. And so it was a cautionary note where we can actually, I think the point here is we actually can pick up human signals of management in white-tailed deer. And this is a very widespread species not of conservation concern. And so when we get to smaller populations, it actually could have much more of a cost. And so just to summarize that again, no evidence of gene flow during speciation, but we can definitely pick up climate and human drivers of demographic change. And collectively, what these two questions have led my group to wonder is, what is keeping them as two distinct species? And, you know, I mentioned that if you were to go in Alberta or Kansas, you will find hybrids. But most of the evidence suggests it stops at F1, mitochondrial aside. And so this begs the question, what is keeping them as two distinct species? They overlap, they exchange, they contemporarily hybridize. What keeps them as distinct species? This is the mitochondrial model. Again, I would argue likely due to incomplete lineage sorting, but there's clearly sharing of mitochondrial genomes. And this has led me to wonder about this idea of maybe there are genomic incompatibilities preventing swarming. So something happens when the two species hybridize. They can exchange mitochondrial genomes, but their nuclear genomes stop. So you can get introgression of mitochondria, but not of nuclear. That's what our data would suggest. And so I've been thinking a lot about incompatibilities. And so there's this long historic model of hybrid incompatibilities referred to as the Djemski-Mueller model. Essentially, this is the idea that you can have interactions between at least two genes that in the hybrid more or less don't work. So in the parental species, the alleles of those two genes work together perfectly well. But in the hybrid, that creates problems, and this could lead to, for example, hybrid sterility. And if this is happening in the nuclear genome, might actually explain what we're seeing. And so I've been interested in seeing if we can identify some level of incompatibilities between the two species of deer. Now, to do this, ideally you would see something like we have in the top right. And so this is, my postdoc lab did a lot of work on crow speciation. So if you've been to Europe, you've probably seen one of these two species, the hooded crow and the carrion crow. Hybrid, they're good species. But when we look at their genomes, they're homogeneous except for 3 regions, that's shown here in red. And so you hope to see this because if you're looking for incompatibilities, you can measure this quite easily when you have 3 divergent regions, and that, that would suggest that this is what's keeping them as species, preventing genetic swarming. However, when we look at the genomes between white-tailed deer and mule deer on the bottom here, it's a complete mess. They're highly diverged. There's no obvious candidates to look at to say, okay, this could be creating the incompatibilities. And so this got me thinking about maybe rather than looking for incompatibilities, which is a complex thing to look for from a modeling perspective, was to look for structural variants. And so these are large-scale rearrangements in the genome. We know that they can actually create problems when it comes to meiosis, for example, and cellular division. If you have mother and father give the offspring 2 dramatically different copies of their DNA, that can actually create problems. And we're seeing a lot now in the literature of wildlife where structural variants are probably quite important in local adaptation. And so this led us to this idea of let's find, thinking of this idea of incompatibilities, let's look for species-specific structural variants. So big changes in the genome of one species, but not another. That might actually hold clues as to what's keeping them as separate species. So we did this again using genomes. These are what structural rearrangements look like on the left. These are big changes, translocations, duplications, inversions. And you can do this with a variety of genomic data. We used what's called short read data, but also long read data, where you can actually sequence, tens of thousands of base pairs long. And the idea here was, let's develop a workflow to identify some of these insertions, deletions, duplications, and inversions that are in one species and not the other. And my PhD student and I developed the most convoluted pipeline of all time, shown here. But the key thing here is, at the end of this pipeline, what you should get, and you can look at the bioRxiv paper, is species-specific variants. So it's compatible with all sorts of data sets, multiple reference genomes. And one of the cool things we did was we accounted for reference bias and false positives due to the method. If you're interested in the details, you can look it up. But the end result here is structural variants that are found in one species and not the other. And so when we applied this to the deer, what we found is between white-tailed deer and mule deer, there were 27 fixed structural variants in one species, not the other. Of those 27 structural variants, coincidentally, they encompassed or impacted 27 genes. And so we can look at those genes and see exactly what's their function. We also observed in these structural variants that were found in one species and not the other, increased number of enhancers. And so this is one way our genomes play around to turn things on and off. 2 of the genes that were impacted showed signs of selection. So we can test for selection. And so it would suggest that they're actually quite important for speciation. And then 2 of the ones that really stuck out to us when it comes to trying to understand what's keeping white-tailed deer and mule deer separate species is we see structural variants that are known to influence sperm cell differentiation and odor recipients. And so if you study deer or most wild animals, smell is a premium. And that is very likely could be potentially maybe a pre-zygotic mechanism keeping them separate. Sperm cell differentiation is interesting because the one study that was done in the 70s did show that hybrids had reduced sperm motility. And so this is one way we're going about trying to understand what's keeping these 2 species separate. structural variants might be the answer. So that's where we're at right now with the story. And one of the things we're doing is we're actually trying to understand recombination as well, and how that might influence speciation through suppressing regions. And so stay tuned for that. So that's the Canada reads portion, a lot, and hopefully tractable. If you don't do genomics, I may have been a bit jargony. But I wanted to also spend a bit of time telling you all about work we're doing on heritability and gene mapping. Especially if you're studying livestock, I think this is relevant. And so if you don't know what heritability is, or if you do, just a refresher, this is a value that lies between 0 and 1. And it's the variation in a trait that's due to genetic variation between individuals in that population. So it's the genetic component or the additive variance. So this equation on the left is narrow sense heritability. The numerator is the genetic component. The denominator is the genetic component, and the environmental component, what we call the phenotypic variation. And so this value will be between 0 and 1, and that's your heritability. How much of the phenotypic variation is due to genetics? In humans, for example, things like height, 80% heritable. So most of our height is due to our genetics. Some cool examples, well, not necessarily cool, but we do know there's also an environmental component. There's the observations during both world wars due to famine. People actually shrunk. But there are traits that are highly heritable, like height, red blood cell phenotypes, and then we have personality traits in humans, less heritable, behavioral complexes that show reduced additive variance. And one way we can go about estimating heritability, and this is what I've been doing, and it's kind of a new method, is relying on genomic data. And so in the livestock world, but even in the wildlife world, we've largely used pedigrees, human world as well. And so to solve this equation here on the left, there's a variety of ways to do it. The animal model is one, but it typically requires a pedigree. It turns out, though, if you estimate a genome relatedness matrix, you can actually solve that equation and come up with meaningful and repeatable estimates of heritability. And so this is the paper here, if you're interested. I won't go into any detail. It's reproducible. It matches human estimates, and in my lab, we're fairly confident in it. But so now you can get away with genomic data from essentially unrelated species or unrelated individuals and estimate heritability. You no longer need to rely on a pedigree. And there's a variety of studies that have shown why this is important. My lab in Alberta, where I did my PhD, showed heritability of horn size and impact of artificial selection by hunters. But this study came out last year that I think is relevant because it showed the additive variance or the genetic component, that there's actually a genetic component to fitness. And so the additive variance, so the genetic contribution to fitness, is non-zero. And we see an example, or we see the variation here on the right. And so it turns out fitness, whether that be number of offspring survival, whatever proxy you use, is heritable. And in this paper, again, this was a Nature or Science paper, they argued that the additive variance, the VA component of fitness, is the most important evolutionary parameter that you could estimate. And so it's really important. And in my world, what I've been interested in is trying to understand the heritability of deer traits. And this is interesting for a couple of reasons, because if you chat with a hunter or a farmer, and I hunt deer, and so I interact with a lot of these folks, one person will say it's the environment, one person will say it's the genetics, one person will say it's a combination. And it turns out in deer, we don't really have a ton of data on the heritability of traits like antlers. The handful of studies that have been done are usually mother offspring or dyads. And we also know in captivity, the VA component in that model is inflated. And so I've been interested in trying to understand the heritability of these traits because, you know, here's Superstar. People are selling semen of this deer for $2,500, presumably because the semen is going to carry the genetics for big antlers. Yet this question has largely not been answered. How heritable is this trait? In addition to finding out if things are heritable, we also want to find the genes underlying these traits. And so typically you can do this with genome-wide association studies, and in the livestock world, in the human world, you can do this because you have hundreds and thousands of samples and lots of money. In the wildlife world, it's much more complicated, and so we've had to come up with some creative ways to do this. And so we're interested in finding also the genes underlying traits like migration and movement, in addition to antlers and body size. If we have time, ask me about this photo on the left here. This photo circulated US Customs for a year before it made its way to my desk, and I'll tell you that story if we have time. And so we've been applying this method of using genome-relatedness matrices to estimate heritability. And in one example, we did this on around 300 deer that were harvested on Anticosti Island off the coast of Quebec. We also estimated heritability of something like hind foot length, which is body size. We would expect this to be highly heritable. This gives us confidence in our method. And so we find, 60% of the variation in hind foot to be due to genetics. And it turns out when we look at antler size, things like spread, how wide they are, and antler beam thickness, very low heritability. And in fact, the only aspect of antlers that we measured that had moderate heritability at roughly 33% was the number of points. So this would suggest antlers in particular have moderate to low heritability. And this is the first study that's ever done that on a free-ranging population. We've also been looking at the heritability of migration. So I've been working in a cool population in Colorado with hundreds of radio-collared females. And the radio-collar data gives us tons of information when they start their migration, when they end it, how long their migration is, how fast it is. And all of these traits we can look at and see, are they heritable or not using this approach. Again, we had our standard metrics, hind foot length, roughly 30% heritable, lower than we would have expected. But again, giving us some confidence in the model. And it turns out, with respect to some of these behaviors with migration, most of them are not heritable, but movement rate is highly heritable, how fast the animals were moving. And we can display this a few ways in our model. So everything in green is the additive component. So the majority of that bar is green for speed, suggesting the majority of that variation is due to genetics, where in blue, that's the effective year. And it turns out when animals start and stop their migration is largely driven by the year, which makes sense. In Ontario right now, we have snow earlier than is typical. It's a year effect. And deer in particular are going to respond to environmental cues. And deer in Ontario in particular, when snow comes, that often initiates their migration. So we expect a high degree of variation in a trait like that, not due to genetics, and we see that. But we see a high level of the movement rate, the speed at which they move seems to be heritable. And it turns out, and this might be, I'm sure someone in the audience might know more than me, but in horses, race horses, this seems to be consistent with what we see in that literature. So we've had to rely on non-wildlife animals to put our research in context. We've also done some cool things to identify the genes underlying these traits that are heritable. And so one thing we did early on, largely due to budgetary reasons, we created pools of animals. And so we had DNA of individuals with big antlers and small antlers. And we had individuals from big bodies and small bodies, and we pooled them together. And so we mixed all of their DNA together. This is called pool sequencing. It's done in Drosophila. One of the cool things here shown on this plot is all things were equal. So they were the same age, but then same aged animals, but different number of points. And so you can see large antlered and small antler, blue and yellow in the top right. On average, they were the same age. So they were from an island. So the only difference between these two individuals was really their antlers or their body size. Pull them together, we look for genomic differences, trying to find the genes underlying these traits, and we can do that. These aren't the nicest plots, but we see peaks in the genomes when we compare them that are consistent with what you would expect if there were alleles that differed between the two pools. And we see that for both body size and antler. And these are definitely candidate genes when it comes to trying to understand what genes are controlling these traits. We did the same thing for migration. Genomically, there's no structure at all. We took a different approach where we identified outliers between migration direction. And so everything in the top part, everything in red is a genetic region that's highly diverged between migratory directions. And then when we compare within migratory directions, there's no divergence. And so again, the only thing that's differing between these two comparisons is the migration direction. And we're pulling out genes that seem to be differentiating between them. And so the genes that seem to be implicated in antler genes often relate to cancer. And we've seen this in proteomic studies and RNA studies. If you're interested in antler growth, it's an incredible process, but it's sort of rapid, uncontrolled, controlled growth. And so they seem to be exploiting, in a few cases, cancer genes. Body size, we get hundreds of genes that make sense. This is a polygenic trait, but insulin growth factor comes out. And I know in dogs, this comes out a fair bit in body size. In migration, we see body fat influencing it and epigenetics. We've also focused on island populations. And we just finished up a study off the coast of Newfoundland on those French islands that you may or may not have heard about, Saint Pierre and Miquelon. They have it's what's in blue and yellow. They introduced a bunch of deer there in the 50s, and it turns out these deer have a high level of piebald, so that's the bottom here, so pigmentation, and then malocclusions, and so jaw deformities. And then the key deer are an endangered species, but also Google key deer if you have time, you'll see they are tiny. So they are miniature deer. And so we have these island populations that are phenotypically divergent and allow us to potentially identify the genes underlying these traits. And it turns out we identified some candidates. For leucism, LAMTOR2 is one of the markers that comes out in all of the animals that has it, that has the trait. And it turns out we know the pathway impacts melanocytes. Malocclusion genes, the big one came out as NPVF, which we know in mice results in craniofacial abnormalities. So we have some high confidence in both of these genes that they're actually maybe not causative, but definitely part of the pathway. Leucism is probably a simple trait with only a handful of genes. And so we are still exploring and trying to validate this. And body size, again, hundreds of genes implicated, but IGF comes out again. So we're able to identify heritability of these traits, but then we're also identifying potential candidate genes. And the goal ultimately with the candidate genes in some ways is to identify biomarkers that we can use for breeders, but also managers to assess, the level of how many carriers there are of these genes that are potentially problematic. Lots of things we're doing in that carry on from this work. We're validating these genes. I'm very interested, if anyone knows anyone that has a piebald deer, we would very much like to validate this gene in a sample that's not from the island. And we're also doing a fair bit of epigenetic work on deer. And we're doing some epigenome-wide association studies with chronic wasting disease, for example. So lots to do. And one thing I wanted to highlight, just to kind of conclude, is when you're doing these quantitative genetic studies and you're interested in traits, a trait like antlers is going to be, well, females don't have antlers, but a trait like body size and it is going to be dependent on sex and age. And so when you're doing these models, you actually should be incorporating sex and age into the animal model, into your ability to estimate heritability. And in wildlife, that's next to impossible. Maybe we get sex, maybe we get age class. And I just wanted to highlight, we've been doing epigenetic work now where we've developed tools. And so on the left here, based off of one methylation site, we can identify age class. And we can also reliably sex animals based off of a single epigenetic marker. And we can actually give you an age in years. And so one of the cool things we're trying to do now in these quantitative genetic models is integrate epigenetics at the very least to estimate sex and age of the individuals. And we're also validating some of these biomarkers using what's called a RAMP assay. And so lots of things on the go. Hopefully that wasn't too much, but I'll take time for questions. Oh,
Sorry,
muted.
I was muted there. Yes. Thank you so much for your presentation. It was really interesting. I'll open the floor to any questions from the audience in person or online. If anyone, Colin, go ahead.
Perfect. Can you hear me there, Aaron?
Yes, yeah, perfect.
Perfect. Thank you for the talk. It's always nice to hear from, obviously, we're very livestock focused here, so it's always nice to hear kind of the other side of things specifically. And I was interested, and please correct me if I misheard on anything specifically, but you were talking about kind of the impact on the whale population historically was overestimated based off the fact that we were using mutation rates that actually weren't specific to that population of whales.
Correct.
Specifically, and so I was wondering what is kind of the ranges across different species that you've seen, because I've done some work with it specifically in cattle, and it's seen that for each individual has between like 30 to 60 new mutations, most of them being benign, a few being recessive, and then very, very few being dominant within those animals. So just curious as to what kind of ranges you've seen across different species.
Can you see my screen still?
No.
Okay, let's again. The variation in mammals isn't super high. So, I mean, you were talking about sort of categories of mutations. Can you see my screen now?
Yes,
or no?
Yes, yeah. I can see your screen now, yeah.
So everything in orange is mammals, and it's more or less 0.5 to 1 10 to the negative 8. And so I'm guessing, depending on, I don't know how you did it. When we did it in deer, so you said 50, we identified, we had 4 trios, we took the average, we see about 40 to 50 de novo mutations, and that is the numerator. The denominator is the genome, and a mammal genome is 3 billion base pairs, but then you prune out a lot. So I can't remember, but it's probably 42 over 1.8 billion. And I would say, like, so there's, if you look at reptiles, right, that's a fairly large variation. In mammals, and what we've actually done in deer is we took the average of all mammals, and it more or less works. So there's not a ton of variation, though. If you do these type of models, like I have here, or these are called SMC plots, these are all dependent on mutation rate. And this one, for example, is recombination rate. Even those slight tweaks in those values will impact these plots considerably.
Okay.
So I would say, like, the absolute mutation rate in mammals, from what I've seen, doesn't vary a ton, but these models are so sensitive to that variation, it does have an impact. And the whaling example is an extreme one, because it was 10 to the negative 7 rather than 10 to the negative 9 or whatever it should have been.
Okay.
And it was, I don't know, I knew one of the authors, but I think they had a good idea going in that the mutation rate was off, in part because kind of like the translocation data we have for deer, the whaling records are pretty detailed. And like they had exact numbers from the ports in the Northwest. And the genetic data with that mutation rate was so much higher than the whaling records. It would indicate they either missed the majority of harvest, or the mutation rate was off, and it ended up being the latter.
Okay, perfect. Thank you.
Does Flavio have a question?
Yes.
Yes, he does. Sorry, I keep muting myself.
I was just waiting the prompt. Thank you so much for the presentation. It was really, really interesting to us here. So I have 2 questions. The first one, you presented a graph with what the effective population size that changed over time.
Yep.
But it's hard to have idea of currently, like nowadays, which is the value of the effective population size? For instance, one of the species. So I like to compare with the cattle, livestock.
Yeah. So that's a good question. I would say do not interpret these literally, and even like so like the SMC authors, if you're like, I think it was the one of the SMC reviews. So these are sequential Markovian coalescent methods. There's an overlooked point in one of, I think it's essentially the big patterns that matter, not the absolute values. So I'll say that when I interpret a plot like, can you see my screen?
Yeah.
Yeah, a plot like this, I'm less interested in the absolute values rather than the patterns. So this is historical NE, and it's very high.
Very high, yeah.
And though in some ways I have to, if I'm making an argument that lineage sorting is why the mitochondrial data is a mess, the only way that explanation works is with a high effective size. And so in some ways, maybe it makes sense. But
so,
yeah.
So why I'm asking this? So you have this very high NE.
Yep.
So we expect then a very low level of linkage disequilibrium in the genome.
Yep.
So what is the density of the markers that we are using, for instance, in your GWAS analysis, to be able to capture the QTLs?
Yeah, so pi, for example, is 0.004 to 0.008. So it's relatively high for wild mammals. What is humans? 0.001 or 0.002 pi. So our density is relatively high. Like that's 4 mutations every 1,000 base pairs. And
yeah, so you are working mostly with the sequence, right? You are sequencing. You don't have like SNP panels.
no,
for
that's right.
years,
It's whole genome sequencing.
Yeah, the whole. And what is the final density of the markers that you use?
If it depends if we prune or not. But again, so a pi value of 0.005 is five mutations every 1,000 base pairs.
Okay, 5 mutated. Okay.
Yeah.
Okay. Yeah.
So it's not like stickleback level, but it's higher than domestic dogs.
Mm-hmm.
So we're missing, yeah. And I'm relying on capturing a large window. I'm not under an illusion that any given mutation is causative. And we've done a lot of GWAS that I don't even show here, environmental associations. I'm relying on capturing like kind of the pool seek stuff. Let me just see what that looks like. Stuff that's hundreds of thousands of base pairs, ideally long, like a big chunk of the genome is being pulled up. That gives me more confidence that that's a real signal than a single mutation.
Yes, to have some indication of a peak, not just
yeah, exactly. Yeah.
So my last question, sorry, I'll let others to ask. You mentioned that we are looking at translocations. So how common is translocations in deer? What is the percentage of animals that you find with the translocations?
Oh, for structural variants?
No, I mean translocation, really translocation across chromosomes, like rearrangement of chromosomes.
So, yeah, when we, it ended up being a bit more methods heavy, but when where am I? If we use long read data, every single individual has it. Has translocations, has structural variants. The short read data is a lot of false positives. So in here, for example, what we had confidence in is, like 8, 9, 10, 16 transversions were fixed between species, but each individual, certainly with long read data, would have it. Like there's tens of thousands of structural variants detected in each one of our long read samples. Is that what you're asking?
Yeah, no, I was thinking more like large translocations that we would see rearrangements of across the chromosomes that you could visualize even with look at the microscope, like
Yeah, so I mean, I would love to work with someone on that because we know, so like, but I don't know the answer. Like, quite a few of them are 30,000, 100,000 base pairs. Would you see that if you did sort of a standard microscope staining? Like, is that big enough to see?
I think so, yeah.
Okay, because that would be cool to do.
Interesting. Why I ask this, because in the livestock species, there is some species that have more than others. Like in swine, depending on the breed, you have up to 3% of large translocations.
Yeah. Like,
yeah, and in the past, they thought, wow, this all translocations are bad, no? But in fact, there is some translocations that are quite favorable for the traits that we select for.
yeah, and
So
I would,
there are
yeah,
big rearrangements across the chromosomes that are beneficial, right?
yeah,
And then, but in principle, the companies would do would analyze the bars, right? In the nucleus for translocation, would discard all translocations, all
yeah.
bars with translocation. But now this has been reevaluated because some translocations are quite positive for the trait. So I was just curious if all these species of mammal would have a,
Like, we know
what would be the level.
like in wildlife, I don't know a ton. Like, I know brocket deer, so they're big in South America. Massive karyotype differences between subpopulations, and we're looking a little bit at if there's geographic segregation of some of these SVs, but we haven't. I don't know. That's a good idea for us to look at, but I haven't seen anything that does it in wildlife, and even just like percentage. But I would be interested in trying to see if we can pull that out of our data.
Yeah, if you search more like the swine literature, you're going to find
You'll see
more
it,
information. Yeah.
Okay, thank you.
Than other livestock species. Oh, thank you so much for the presentation, for the
No,
answer
thank
the
you.
questions. Yeah.
All right. Thank you for the great discussion, everyone. Unfortunately, we are out of time. But thank you again, Dr. Shafer, for your presentation. It was very interesting. And we will be in touch when we post the recording of the seminar onto the YouTube channel. And I'd like to thank everyone for joining us for the last seminar of the year. So enjoy the holidays, and we will be seeing everyone in January. Thank you.
Thanks, everyone.
Thank you. Take care.
Thank you, Gabby.